Dear friends, let's understand the practical exposure and uses of microscopes. Today, we will learn about microscopes and its applications. As we know that on the basis of illuminating source, mainly three of two types, electron and light microscopes, electron microscopes uses electron as a source of illumination. The electron microscope is one of the most advanced microscopes used today. This microscope is powered by a beam of electrons with very short wavelengths. These electrons strike objects that come into its path and decrease the resolution of the microscope. The electron microscope is one of the types of microscopes used to study cells like tiny viral cells as well as large molecules. There are different types of electron microscopes. Let us collect a brief idea. Transmission electron microscope. The transmission electron microscope is used to study cells. Ultra-thin slices of microorganisms like viruses are placed on a wire grid. Then these cells are stained with salts of uranium or lead and then used to observe under a transmission electron microscope. The electron beam is scattered by the electron densely coated parts of the cells and the image is observed on dark and light background as stained by heavy metals. Scanning electron microscope. Scanning electron microscope is also a type of electron microscope with lower magnification power than a transmission electron microscope. However, this microscope helps in viewing three-dimensional images of microorganisms and other specimens. Gold and palladium is used to coat the surface of the specimens mounted on a scanning electron microscope. Light microscopes. Now, let us understand the meaning of light microscope. It is a type of microscope which uses light as a source of illumination. Several types of light microscopy are used today in laboratories which are working on the different pattern of illuminating which are based on the types of specimens. These include as bright field to observe transparent thin specimens, stained by stains interference and differential interference contrast microscope to observe transparent but thick specimen. Dark field microscope is used to observe semi-transparent or semi-opaque or translucent specimen. Opaque specimens are observed using reflection mode and transparent fluorescent specimens are observed using fluorescent microscopes, etc. and many more depending upon the purpose of observation and uses. Let us discuss an introductory idea about these basic types. Bright field microscopy. This is the commonly used or routine microscope. In bright field microscopy, the field of view is brightly lit so that organisms and other structures are visible against it because of the different densities. It is mainly used in stain preparations. Differential staining may be used depending on the properties of different structures and organisms. This may be simple or a compound microscope. The other way of permanent specimen is fixed, dehydrated, cleared, embedded in paraffin wax and sectioned. The ribbon of section is marked stained, processed and covered with Dipiax or Canada balsam and then covered glass-lipped. Simple microscope. A simple microscope or single lens microscope uses just one lens for magnification of the sample. It is the original light microscope that is now considered primitive. It uses a single convex lens that is commonly observed in the loop. it is generally used for gross or whole mount small organisms or tissue sections. Compound microscope. The most commonly used microscope and the integral part of a school or college lab is the compound microscope. This compound microscope uses two optical parts called the eyepiece and the objective lens. The compound microscope can provide up to 1000 times magnification with photography zooming. Specimens are processed to prepare thin layer as single cell layer or thin sections or smear or squash or the plain glass slide. Two different methods are used. One for temporary, in which the thin specimen is stained and mounted on slide merged in mounting medium, or glycerin water and covered with cover slip. Stereo microscope. Dissecting microscope or stereo microscopes have low magnification power. This is also the type of light microscopes that help in observing specimens that are larger in size. It has reflection mode of illumination. It contains two optical paths that are at different angles and help the user see the specimen in 3 dimensions. The stereo dissecting microscopes and the types of microscopes used in anatomy and physiology for carrying out microsurgery, dissection, fine repair, sorting, as well as forensics. The advantage of dissecting microscope is that it can be used on living specimens, but it has a low magnification power. Dark field microscopy. In dark field microscopy, the field of view is dark and the organisms are illuminated. A spatial condenser is used which causes light to transmit to the specimen at an angle not oriented towards microscopic path. It is used for observing bacteria such as Treponemas, which causes syphilis, and leptospirosis, which causes leptospirosis, and other semi-opaque to translucent specimens. Phase contrast microscopy. Phase contrast microscopy allows the examination of live unstained organisms. For phase contrast microscopy, spatial condensers and objectives are used. A phase plate is placed at back focal plane of an objective and an annular diaphragm is placed at front focal plane of condenser. These alter the phase relationships of the altered order and zero order light passing through the object and that passing around it. Fluorescent microscopy. In fluorescent microscopy, specimens are stained with fluorochromes or fluorochrome dyes. Light of high energy or short wavelengths from halogen lamps or mercury vapor lamps is then used to excite structures or molecules within the specimen or dye molecules attached to it. These excited molecules emit light of different longer wavelengths, often of brilliant colors. Auramine differential staining for acid-fast bacilli is one application of the technique. Rapid diagnostic kits have been developed using fluorescent antibodies for identifying many pathogens. Remember, microscopes can be made more sophisticated with additional accessories for ease and superiority in functioning. Here, in this practical approach, we will specifically focus onto light microscopes, especially a routine compound microscope that we use in our laboratory to observe biological objects having micrometric dimension or micrometric structure of higher organisms, that is, they may be bacteria, algae, fungi, yeasts, or simply a eukaryotic plant or animal cell, and sometimes cell organelles as well. Components and function of compound microscope. The main parts of compound microscope consists of basically 3 units. It includes illuminating system, optical system, and the third, accessory supplementary system and documentation. Let us go through it in detail along with their individual functions. Illuminating system has following parts. Built-in light sources. An illuminator is built into the base of the microscope. A halogen bulb provides the best illumination. Halogen lamps are low wattage, high intensity lamps and are the preferred light source. Though costlier, these have the following advantages over tungsten lamps. They emit white light. They have higher luminosity. They have compact filament and have longer life. On the top of the illuminator is an inbuilt filter holder to fit the filter of desired quality. Two-sided mirror. The two-sided mirror provides necessary illumination through reflection of natural or artificial light. It has two surfaces, one plane for artificial light and other concave for natural light. It is supported on two sides by a fork fixed on a mount in a way that permits free rotation. Condenser. The condenser illuminates the specimen and controls the amount of light and contrast. There are 3 different types of condensers. Some condensers have a rack and pinion mechanism for up and down adjustment. The numerical aperture of a condenser should be equal to or greater than that of the objective with maximum numerical aperture. An iris diaphragm is provided below the condenser. This adjusts the numerical aperture of the condenser using objective having low magnifying power. A swing out type filter holder may be fitted above or under the condenser. In some microscopes, the filter holder may not be swing out type. The filter holder holds detachable filters when required. Condenser centering screws, when present, are used to align the condenser with the objective. A condenser raising knob may be present or the distance may be fixed. Filters. Blue filters are used to change the light from ordinary electric bulbs into a more natural white light. The neutral density filters are used to reduce brightness without changing the color of the background. Grain filters may be useful in some situations. Optical system has following parts. It includes objectives. The image of the specimen first passes through the objective. Objectives with magnifying power 4x, 10x, 40x and 100x are commonly used. The magnifying power is marked on the lenses and is usually colored code for easy identification. The numerical aperture is the measure of light gathering power of a lens. The numerical aperture corresponding to the various magnifying powers of the objective is different. That is, for 10x it equals to 0.25, for 40x it equals to 0.65 and similarly for 100x it is 1.25. A high numerical aperture indicates a high resolution power and thus useful magnification. To provide the best image at high magnification, immersion oil is placed between the slide and the oil immersion objectives, especially only in 100x. Unlike air, immersion oil has the same refractive index as glass. Therefore, it improves the quality of the image. If immersion oil is not used, the image appears blurred or hazy. Eyepieces. The specimen is viewed through the eyepiece. It has a lenses which magnifies the image formed by the objective. The magnifying power of the eyepiece is in the range from 5 to 20x. A movable pointer may be attached to the inside of the eyepiece. In binocular microscopes, the two eyepieces can be moved closer or farther apart to adjust for the distance between eyes by pulling, pushing, motion, or by moving a nerd ring. Accessory and supplementary system along with documentation includes the microscope tube known as body tube. The microscope tube is attached on top of the arm. It can be of the monocular or binocular type. It supports the eyepiece on the upper end. Mechanical tube length is the distance between the place where the object is inserted and the top of the draw tube into which the eyepieces fit. In modern microscopes, it is not tubular. It contains prism that bend the light coming up, thus providing a comfortable viewing angle. In a binocular tube, the light is also split and sent to both eyepieces. Nose piece. The nose piece is attached under the arm of the microscope tube. The nose piece houses the objectives and rotates them. The objectives are arranged in sequential order of their magnifying power, from lower to higher. This helps to prevent the immersion oil from getting into the intermediate objectives. Mechanical stage. Mechanical stage holds the slide and allows it to be moved to the left, right, forward or backward by rotating the knobs. It is fitted with fine vernier graduations as on a ruler. This helps in relocating a specific field of examination. Coarse and fine focusing knobs. The coarse and a fine focusing knobs are used to change the distance between the specimen slide and the objective. The coarse focusing knob alters this distance rapidly and is used to bring the specimen into the field of view using an objective having low magnification power. The fine focusing knob changes the distance very slowly and permits better viewing of the object. One revolution of the fine focusing knob should gradually move the mechanical stage by 100 micrometer. The movement should be smooth and free from jerks. Stage rack. The slide holder is used to secure your slide in place so you will not inadvertently change its position. The paired knobs below the stage on the right hand side move the slide holder either forward and backward or left and right. These allow you to precisely control the position of the sample on the stage. The scales on the slide holder can be used to determine the position of an objective on the slide. Thus, we can find an object, note its location, and move to a new position and then later turn to the mapped object. Camera lucida. A camera lucida is an optical device used as a drawing aid by artists. The camera lucida performs an optical superimposition of the subject or a specimen being viewed upon the surface upon which the artist is drawing. The artist sees both scene specimen and drawing surface simultaneously, as in a photographic double exposure. This allows the artist to duplicate key points of the specimen on the drawing surface. thus aiding in the accurate rendering of perspective. Functioning of compound microscope. There are three main optical pieces in the compound light microscope. All three are essential for a sharp and clear image. These are condenser, objectives, and eyepieces. The condenser illuminates the object by converging a parallel beam of a light on it from a built-in or a natural source. The objective forms a magnified, inverted, upside-down image of the object. The eyepiece magnifies the image formed by the objective. This image is formed below the plane of the slide. The total magnification of the microscope is the product of the magnifying powers of the objective and eyepiece. For example, if the magnifying power of the eyepiece is 10 times and that of the objective is 100 times, then the total magnification of the compound light microscope is 10x to 100x, which gives 1000-fold magnification. Routine operation of the compound microscope. Ensure that the voltage supply in the laboratory corresponds to that permitted for the microscope. Use a voltage protection device if necessary. Turn on the light source of the microscope. With the light intensity knob, decrease the light while using the low magnification objective. Place a specimen slide on the stage. Make sure the slide is not placed upside down. Secure the slide to the slide holder of the mechanical stage. Rotate the nose piece to the 10x objective and raise the stage to its maximum. Move the stage with the adjustment knob to bring the desired section of the slide into the field of view. Focus the specimen under 10x objective using the coarse focusing knob and lowering the stage. Make sure the condenser is almost at its top position. Center the condenser using condenser centering screws if these are provided in the microscope. Open the condenser iris diaphragm to 70 to 80% to adjust the contrast so that the field is evenly lighted. Adjust the interpupillar distance till the right and the left images become one. Focus the image with the right eye by looking into the right eyepiece and turning the focusing knob. Focus the image with the left eye by looking into the left eyepiece by turning the diopter ring. Put 1 drop of immersion oil on the specimen. Change to 100x objective. Increase the light by turning the intensity knob until a bright but comfortable illumination is achieved. Focus the specimen by turning the fine focusing knob. When the reading or observation has been recorded, rotate the objective away from the slide. Release the tension of the slide holder and remove the slide. If immersion oil was used, wipe it from the objective with lens paper or muslin cloth at the end of each session of use. In general, avoid wiping the objective except when it seems to be dirty. Turn off the light. Cover the microscope when not in use and take necessary precautions against fungus. Applications of microscopes. Applications of microscope are enormous. Remember, type of microscopes is selected based upon and their uses and our purpose. To know the applications, it is mandatory to divide applications into sectors. for instance, applications in specific biological science and non-biological sciences, which includes physical, chemical, material, or mathematical science. Applications of microscope in biological sciences. In biological sciences, unless further categorizing sectors, it is not clear to know its application. It has applications in various disciplines, as in microbiology, to study microscopic organisms, for example, protista, prokaryotes, archaea, viruses, algae, fungi, etc. Cytobiology, to study cell biology, for example, cell structure and organelles therein. Clinical biochemistry, to study blood count, performed in clinical laboratories using microscopes to count the relative frequencies of erythrocytes, white blood cells, platelets, etc. Histology, to study tissue morphologies. In taxonomy, it is used to identify microscopic morphological differences between related species. Forensic pathology, used to diagnose causes of death, especially when death appears to be traumatic. Forensic botany, it is used to identify minute plant matter at crime scenes in order to determine where the crime victim was killed. Forensic entomology, it is used to determine time of death in decomposed corpse by examining insect larvae. Paleontology, to identify minuscule plants and animal remains such as pollen grains in fossil sites. Archaeology, to identify small particulate matter associated with ancient human settlements, including ingested foods, vermin, that is mites, and traces of domesticated animals. Applications of microscopes other than biological sciences. In physics, to understand the size and structural topography if a minute crystal matter. Chemical formulations are subjected to know their structural particulate size, property, and nature through microscope. Material sciences, geology, and mathematics are largely dependent on the preliminary data of knowing the structural configuration and particulate size of natural and or synthesized materials, either to know or match the matter structure. Indirect evaluation of matter mass can also be achieved after calculation using mathematical formula after knowing the measuring the exact size, shape, volume, and topography surface is also of importance when a minute body or a matter is to be envisaged. Summary. Friends, now it's a time for us to summarize what we learned. We have studied various types of microscope according to their purposes and constructions. We studied here in detail of construction, including the mechanism, functioning, including the main parts, and the principle of magnification along with resolution, operating and handling of compound microscope, and also the applications as well about various microscopes. Applications include in both biological sciences and apart from biological sciences, including physics, chemistry, archaeology, material science, and mathematics. At last, we also learned the individual purpose of various types of microscopes, where and when to use. Thanks.